Great. So today we'll just start off with welcome and announcements. Scott has a list of recent and upcoming events to highlight. We're gonna try out a breakout room activity where we'll put 2 to 3 people in a breakout room and just have you all have a conversation about what you're looking forward to this summer, what were your favorite observations for the past year, and then come together as a full group and post in the chat kind of what you talked about. We're going to spend some time reviewing the Vital Sign Key Messages update, discussing how you thought that revision process went and considerations for the next update. And we have 2 presentations. One, the distribution of abundance of sea lions in the inland waters of Washington by Tom Jefferson. And then the Southern Resident Killer Whale Remote Health Metrics presented by Hendrik Nollens. We'll do a brief break and come back, review the work plan update, the 2023-2024, talk through that, and then Scott will be leading us through a marine mammal work group species walkthrough, kind of highlighting the purpose and recent accomplishments of the subcommittees, and highlighting some upcoming needs as well. And also opening the floor to any work group members who want to highlight projects or upcoming funding proposals. Any questions on the agenda before we move on here? Great, so I'm going to hand it off to you, Scott, for the recent events and upcoming.
Great. So I wanted to just highlight one recent event was, thanks to contributions from many, I was able to represent us at the Marine Waters Report Workshop, which happened last week. There were about 25 folks mostly from the Marine Waters work group, but then good representation from the Salmonid work group and the Forage Fish work group. So it's very interesting. And I will provide a link to the slide deck for that. Is it okay if I just paste these into the chat?
Absolutely.
Yeah, I'll just put them in here, but these will be provided in the notes from this call. Oh, we're not going to get the links. It's all right. So there's a slide deck. I basically went through 2022 marine mammal conditions, trends, and statuses. And then I guess there are 2 upcoming events I wanted to highlight. One is on the 13th. Brad Hansen is presenting on harbor porpoises. And then the next winter, there'll be another set of pulse presentations, including talks on killer whales and sea otters. So that'll be next October, November. Since we don't meet during the summer, I just wanted to flag those. In the longer run, we'll provide links to Orca Month events, and just wanted to encourage folks to not only attend those, but also there's still time to interact with Orca Network and other groups that are coordinating those events and volunteer to participate as monitoring experts. I also wanted to share from Jared that they've been running webinars this last winter, and they are often quite informative. They're aimed at a more of a recreational boater and broader community scientist audience, but I'm finding them to be quite informative in terms of getting some perspective on what's happening up in Johnstone Strait and the Strait of Georgia. Yeah, and then we'll provide a link to, there's not a lot happening this odd year, but in 2024, there'll be conferences for the Marine Mammal Society and the American Cetacean Society and the Acoustic Society of America meeting is coming up this next fall in Australia. So I think that's everything. While we have you, though, if you're still with us, Andrew, are you planning on doing the Marine Mammal Symposium next fall like you did last fall?
So yes, we are planning to hold it. It'll be a slight different running it because I'm on sabbatical this year, and if things go right, I'll be on the Galapagos Islands for all fall. And so, but we are setting up a system to run it this fall. And that's always done the last weekend in November.
Super. Okay. Yeah, I'll pass it back to you, Nicole, but also ask if anybody else knows of events that we've missed. Those are the ones that I was going to flag.
Thanks, Scott. Yeah, I wanted to ask this group if there's any other upcoming events that we should flag in our meeting summary to distribute to the wider work group list that couldn't all attend today. Opening the floor if anyone has additional events they want to flag. All right. Well, it sounds cool. Oh,
I got one.
Yeah.
The Ecosystem Recovery Board is being run in Friday Harbor on the 18th of May. So coming up, and I know southern resident killer whales are on the agenda, as well as oil spill prevention, derelict vessels, and vessel management to protect marine resources. So all items that have implications for marine mammal conservation. And that will be held at the Grange in Friday Harbor. And it will also be streamed live on TVW.
Yeah, and in preparation for that, I'm hoping to wrap up a summary of Southern Resident Killer Whale Task Force related budget items and new laws that have passed. And if this group would like a copy of my summary, I'm happy to send it to folks. A lot of salmon related stuff in there too.
thanks, Tara. Alejandro?
Yeah, just quickly, I forgot to mention that the Norway student chapter of the Society for Marine Mammalogy is hosting their annual conference, actually this weekend. I believe it's in Newport. I know it's in Oregon State. So everybody's welcome. I realize it's super late notice, but I just want to flag it for next year. They happen usually early in May. They're hosted by universities, usually University of British Columbia, University of Washington, Western Washington University, or Oregon State. And it's a great place to meet new young scientists who are doing really cool stuff that we've seen a couple of years later published. So I just want to flag it out, mostly as I mentioned, for next year to keep it in our radar, but it's happening this weekend.
Thanks, Alejandro. Andrew?
Yeah, and I can confirm that it is this weekend. I think I've got 6 or 7 students from UBC driving down, but a long trip from Vancouver. And it is a great event for the students and for them to get together, and you get to hear the latest research and what they're doing. I want to mention one other thing, and Scott, you may have touched on, I'm not sure, but there's talk of a workshop at the end of October or beginning of November on southern resident killer whales, sponsored by DFO and NIMS. And they don't have any dates yet, but the idea is to have people come and present results of all the data they've been collecting. And there's a lot of people with permits, a lot of data being collected. And so far there's just talk of it happening, but stand by for more than that.
Yeah, that's great, Andrew. So we don't have folks on here who are doing drone-based work, but one of the things that is something we'll try to talk about next year is thinking about body condition as an ancillary measure within the vital signs for Puget Sound. So that might be a really nice stepping stone to understanding like what we could measure together across the border. So thank you for flagging that, and I'll make sure that Holly and John and others know about it. I'm sure obviously they'll be there, but we'll talk about it from that context.
And they were on the call yesterday when the workshop was discussed.
Great. Cool. Thank you.
Okay, great. Are there any other announcements that folks want to share? Otherwise, if not, we'll transition to this 5-minute breakout room activity. And we'll keep an eye on folks coming in, but essentially what will happen, we'll put you in groups of 3 to 4. We'll have 5 minutes and then we'll come back as a full group. And, you know, we're kind of a small group right now, so it might be great to just take a few minutes to share out as a full group as well. But want to create some space for conversations amongst all of you colleagues. So we're going to do a breakout room. All right. And we'll be back here in about 5 minutes. And I'll post the prompt, or Scott, it looks like maybe you posted the prompt.
Yep.
In the chat. Great.
Yep. Gotcha.
Okay, wonderful. All right. See you back here in 5. Welcome back. I hope the breakout rooms went well. Let us, any feedback. If you think it was a little choppy, feel free to provide that feedback to me. It's just a way to get folks talking to each other in this virtual setting.
I thought it was good. I mean, it's kind of fun to get to know people a little more personally in a short couple minutes. Hey, Alejandro.
Wonderful. Okay, and please feel free to put in chat in response to that prompt anything you want captured in the meeting summary, circulated to the wider marine mammal work group, or folks that weren't in your group. I think we'll be moving on to the next agenda item, but yeah, please use the chat to continue talking about exciting work that y'all are doing this summer. All right, moving on to the next agenda item, the vital sign key update. So wanted to share with everyone that we went through a revision process through the Marine Mammal Workgroup listserv using the previous iteration of the key messages as a base and then updating them based on Marine Mammal Workgroup feedback and edits. So and I'll put the link in the chat here, the website, the Orca's Vital Sign website has been updated. And what I wanted to hear back from folks is any reflections on the process. There is still opportunity to revise or do some other edits on the key vital sign messages for folks that weren't able to provide those edits. We just wanted to set a time frame so we could get them updated to this point. But as a reminder, these will be tied into the State of Puget Sound report, which is published every 2 years. So the partnership is looking at the update process, not necessarily as annual, which I put in this prompt, but every 2 years. So that being said, are there any reflections or feedback you have on the revision process that you would want us to consider moving forward? We're thinking of using a similar approach for the work plan, which we'll discuss later. So looking for folks to either raise your hand or jump right on in here with feedback on this process.
I'll just say real quick, I just was enjoyed being able to be part of it, which I don't think I normally would have been in the grand scheme of things. So being able to voice a less orca oriented person, being able to help with that was really nice and really easy to do through the Google stuff. I appreciated it.
Thank you, Cindy. Tara or Tara?
Yeah,
sorry.
I was going to say something similar, Cindy. I appreciated the collaborative approach. I think the last time this was done, I think the partnership took the lead on it. And since they help own the vital sign, I was just wondering I guess this is more of a question. So we're doing this revision, and then does it get passed to the Puget Sound Partnership to sort of do the final buy-off, and then we'll see any changes or iterations that they want to make?
Yeah, and it was sent to Natalie and Mary, as well as Katrina, and I didn't get feedback on any things that they would like to see updated. So if you go to that link I put in chat, it's a direct,
Okay.
yeah.
I think
directly
Todd Haas, who was on the call earlier, took the lead on it last time, and I know he's been out of the office for the last couple weeks, so he would be a good person to check in with too.
Yeah, absolutely. And like I said, for anyone who's been out of the loop, or they want to review and provide any additional edits, that is an opportunity that I'll flag in a follow-up email to this group after the meeting, knowing that whatever we land on will be incorporated into the State of the Puget Sound report. So if there are critical flags that you see in this draft that you would like updated, that's something we're really looking for and welcome that additional input. But really appreciate everyone who was able to provide such in-depth edits, and there's still a couple considerations on some comments that were put in. So I'm definitely gonna follow up with an email, just laying out another timeline of potential feedback to be integrated into the final vital sign update for the year.
I'll just contextualize this by saying, like, I think it was much more feedback than we got in previous iterations, where, you know, Lynn and Natalie would sort of iterate with a random selection of other members. And this was a much better process in terms of getting input from the broader marine work group. So hats off to you, Nicole, for coming up with a better process, I think. I'm curious to know whether the regional office or Lynn thought that it worked better, but certainly did from my perspective.
Yeah. Thanks, Scott. Any other reflections before we move on to our first presentation?
Nicole, I just had a question about the occupancy indicator. What are
Yes.
things with that? Is there a group compiling data to do similar to what was done with the numbers indicator?
Yeah, so at this stage, we had the Orca occupancy workshops, and some initial recommendations were sent out prior to the monitoring to accelerate recovery proposal deadline. And what is being finalized in the next week or so is a scoping report. That is basically the outcomes of those two workshops. And then the next step is to be determined as far as really having data for that indicator. But the scoping report hopes to set the stage and provide recommendations to doing that. So we'll be sending that out through the listserv when it's finalized. Great question.
Andrew, you might want to reach out to Brad. I know he's wrote letters of support for at least two proposals. This most recent biennial funding had that indicator and further development of it as in its purview. So I think there's some new ideas and subsets of the transboundary data that ideally could be used to compute occupancy, but it's not a fully solved problem. And my crystal ball says it'll be a couple years before we have, it kind of depends on the geographic scope of the metric, but we have some work to do. And I think maybe we can learn a little bit from Hendrik and Joe today about how to share data better, but I think we're making progress towards it. I'm not sure it's a solved problem yet, though.
Thanks, Scott. Any other last minute question or comments before I tee up Tom for his presentation? And feel free again to use chat. So we have Tom Jefferson presenting on the distribution and abundance of sea lions in the inland waters of Washington. And Tom, I made you a co-host, so you should be able to share your screen.
Okay, sounds good. Let me figure out how to do that. It's been a while.
Okay.
Done this. I think I got it here. Okay, you should be able to see one of my slides there. I should be able to see the title slide soon. Can everybody see the title slide there?
Yeah.
Distribution of
Yes.
abundance of sea lions. Okay.
Yes.
Okay, sounds good. Well, good afternoon, everybody. It's a pleasure to be here, if only virtually, if not actually in person. I guess probably most of us are scattered in various places around the country or maybe around the world. The modern world works that way, I guess, these days. One of the good things about COVID, I suppose. But today I'm going to be presenting some work that we've been doing, looking at the distribution and abundance of the 2 species of sea lions in the inland waters of Washington, including Puget Sound. And this study, the data were collected from 2013 to 2016. And I want to thank my co-authors and acknowledge them, Marius Maltea, who I believe is on the call today, and Eric Ward, who I think is in the field and was not able to be here today. But obviously they were very heavily and actively involved in this work, and I want to acknowledge them for their participation as well. So we'll move on here. First question or first thing to cover is, why are we doing this? So a little bit of background, which probably most of you know. 2 species of sea lions found in inland Washington waters, the Steller and California sea lions. Both of these, of course, are major fish predators, among other types of prey, including, as we all know, several threatened and endangered stocks of salmonids. Sea lions in inland Washington waters were reduced by predator control programs in the 1900s in both Canada and the US. And stocks obviously have rebounded since the 1970s, when protection in both countries was instituted. But unfortunately, despite that, there appear to be no available, or at least updated, empirical estimates of sea lion abundance for inland Washington. What we have primarily is incomplete data from counts in various places, and some sort of guesstimates, I would call them, of how many sea lions might be in the area overall, but we don't really have good recent empirical estimates of abundance. So that's what we were focusing on trying to do here in this study. A little bit more background. For some reason my slide is not advancing. There we go. So as you know, generally pinniped abundance is estimated using counts at colonies when the animals are on land, and then oftentimes making a correction for those animals that are in the water. Distance sampling methods, including line transect, have been used in the past a bit for pinniped abundance estimation, although it's not the major method. But oftentimes they don't correct for animals that are missed on the track line during surveys. And even when they do, they generally only give in-water estimates and not full stock or population size estimates, as it's usually the case that some of the animals are on shore at all times. And of course, that number can vary greatly throughout the day and throughout seasons as well. So our approach here was to use line transect sampling methods from data that were collected in 2013 to 2016 to estimate the abundance of both species of sea lions in inland Washington, and hopefully come up with total estimates that include both the in-water and the on-land component. And these surveys were done, as I said, quite a long time ago now, almost 10 years ago starting. And so this project uses historical data. And it's important to recognize that the surveys were originally set up primarily with a focus on harbor porpoise. And so that was kind of the way we set up our transect lines. But we did collect information and data on all species of marine mammals observed, including the 2 sea lion species and other pinnipeds as well. And so we were able to go back and use these data in a retroactive way to try to look at sea lion abundance. So a little bit about our methods. The map on the right shows the map of the study area, which of course includes not just Puget Sound, but other inland Washington waters. all the way basically to the BC border. The survey, as I mentioned, again, was targeted primarily on harbor porpoises, but we did collect data on all different species of marine mammals. The surveys were flown with systematic transects using fixed wing aircraft. There were a total of 39,399 kilometers of aerial survey effort conducted throughout the 4-year period, 3.5-year period there. So we had quite a large sample size of effort data, and a total of 255 groups of sea lions were sighted during the surveys. And you can see our divisions of the strata that we use for the estimates shown on that map there. And the red outlined areas on the map, you'll see 4 of them there. Those are no-fly zones, usually around naval bases and, or for instance, airports. And we were not able to fly over those areas. So we do have some pretty small scale gaps in those 4 no-fly areas. So line transect methods use, this is basically a standard line transect equations you can see here on the left. We use line transect methods, including both conventional distance sampling and multiple covariate distance sampling. We attempted to use Beaufort sea state as our primary covariate, having an impact on, you know, how sightability or how sightable the animals would be. And the data were filtered. So we only use data collected on systematic transect lines, data that were from transits or connector lines, even though we did record sea lions on those lines. Those data were excluded from this analysis. We only use data collected in what we considered good sighting conditions, which we defined as Beaufort 0 through 2 states with no whitecaps. Only data during effort periods without significant glare issues, and only sightings of sea lions in water or on buoys were included. So any sea lions that were hauled out on land, on islands or mainland areas were excluded from this analysis. Well, not excluded, but they were basically not the data from those were not used in calculating abundance estimates. So we used tagging data to try to get our estimates of the proportion of time that sea lions were spending on land versus in the water. So we incorporated tagging data that had been done previously for studies that had other goals basically in mind. And we used these to develop correction factors for both species of sea lions to get estimates both of animals that we missed, that were in the water that we missed during the survey as the airplane flew overhead, as well as those that were on shore during the time that the surveys were done that we would have missed because they were on land. So for California sea lions, the data came from 8 adult males tagged in Shoal Bay from 95 to 2000. The stellar sea lion tagging data came from 25 tagged animals of different age and sex classes that were tagged in the Strait of Georgia in BC, a little bit north of our study area, but quite close by, from 2004 to 06. And the tagging data were modeled to provide estimates of proportions diving and hauled out. And this is very similar, not exactly the same, but very similar to the approach that we had used previously for doing the same kind of work with harbor seals from the same set of surveys. The availability modeling was done separately for the 2 species. For California sea lions, we only had data available for the spring months. So we assumed no seasonal differences throughout the year. And for stellar sea lions, we had data available from January through March. So we assumed constant availability for the non-breeding months. And basically, we took a similar approach here that we used with harbor seals, using the raw tagging data and statistical modeling and making use of generalized additive models. We cautioned that several assumptions were needed to be made in this study. In particular, because we didn't have sea lion tagging data from throughout all months of the year, we needed to make some assumptions that sometimes the tagging data from a particular season was representative of other seasons in which we had little or no data. So that is definitely a shortcoming of the study. It may present some bias in the results. We hope that they aren't significant biases, but it's something that obviously in future studies would be important to try to resolve that issue. So first of all, just distribution, just looking at the locations of where the 2 species were seen. In the left-hand graph, you can see the California sea lion sightings shown there. Keep in mind that we had much more extensive effort actually in Puget Sound than in the northern waters of the Strait of Juan de Fuca and around the San Juan Islands. So what looks like potentially higher densities in Puget Sound is not necessarily real because, again, these maps are just showing the location of sightings and there's no effort information incorporated into this. So you can't really use these to determine or infer densities. But looking at the seasonal distribution, California sea lions were seen in fairly good numbers during the 3 main seasons where they're in the inland Washington waters, the winter, spring, and fall seasons. There were a couple of sightings in the summer, but there were only, I think, 3 of them, something like 3 or 4, very small number. The graph on the right shows the stellar sea lion sightings, and you can see what appears to be a very strong peak in the fall season or the autumn season. Those blue dots there. There were some sightings in the spring and some in the winter, but much lower numbers, and none in the summer at all. So going into our line transect results, after filtering the data, we used 7,840 kilometers of survey effort and 165 sea lion sightings in the line transect analysis. Data were truncated. You can see on the right the graph of the sightings, perpendicular distance in relation to sightability. We truncated the data at 520 meters, and the final results used conventional distance sampling, no covariates, with a hazard rate model and a cosine adjustment. And you can see a pretty good fit of the model to the data there on the right. Going into the correction factors, proportion available values were calculated for both species. We came up with an estimate of 0.2 for California sea lions and 0.43 for stellar sea lions. And you can see on the left-hand side there, the graphs that show proportion of animals hauled out in relation to time of day on the top and proportion of animals diving on the bottom. And you can see that basically they're pretty much inverse functions of each other. So the maximum number of sea lions were hauled out during the nighttime hours, with minimum numbers hauled out in the early morning. And then of course, the exact opposite pattern for diving sea lions on the bottom. The resulting G of 0 or track line detection probability estimates were 4.98 for California sea lions and 2.30 for stellar sea lions. And here are our estimates of abundance. The upper part of the graph, the upper numbers up here, show the estimates for different areas just for the number of sea lions that were estimated in the water. And what you really want to focus on down here on the lower part is the estimates for the different areas for sea lions, including the correction factor for the number of animals that we estimated to be on land and unavailable because they were diving. So these are our estimates of total abundance for those particular seasons in those areas. And I've highlighted those abundance estimates in the yellow columns here. On the left is California sea lions here, and on the right is stellar sea lions here. And so I just want to bring your attention to the fact that the majority of California sea lion numbers appear to be primarily in the northern inland waters, but with pretty significant numbers in Puget Sound in all the 3 main seasons, and even some numbers in summer. We estimated 33 California sea lions in Puget Sound and Hood Canal in summer. Keep in mind that we do have data for all 4 seasons from Puget Sound and Hood Canal because those surveys were done year round. But our surveys in the northern inland waters and the Strait of Juan de Fuca only occurred in particular seasons and with much lower effort. So we weren't able to produce estimates of abundance for those other seasons in those other areas. If you look at stellar sea lion numbers on the right here, overall the numbers appear to be much, much lower. But again, we don't have information for northern inland waters except for the spring months. And for the Strait of Juan de Fuca, we only really have information for the autumn months. So we were only able to do a few seasonal estimates there. So this approach allows us to correct for unavailable animals without the need to conduct independent observer experiments or multiple platform studies. Our methods take sort of a reversed approach from the way that traditionally pinniped estimates are done, whereby aerial survey counts of, or in this case, we used aerial survey counts of sea lions and corrected for animals that were in the water and hauled out. Traditional pinniped abundance estimation methods generally target a single species. And one of the advantages of this method is that the surveys were done with data collected for all different species of not just pinnipeds, but marine mammals. And so we can use the same basic line transect data set for getting estimates of any of the species that we feel like we have adequate sample sizes for. Obviously, there were a lot of species of cetaceans seen that were seen in very small numbers. And so we don't feel we can do reliable estimates for them. But for any species that we have a decent sample size for, we feel we can, use these data to produce estimates of abundance. And again, I want to caution that due to data limitations, we were not able to estimate abundance for all areas and all seasons. And we did have to make several assumptions about the representativeness of the data for different seasons, where we only had tagging data for certain seasons for sea lions. So we considered that these estimates should be considered preliminary and sort of a first step to getting us on a path to be able to come up with better estimates of abundance for these species, hopefully in the future. They do provide, as far as we're aware, the first real empirical estimates of abundance for either species of sea lion in inland Washington waters, certainly at least for relatively recent times. And the project does show or indicate that there are several thousand California sea lions present during at least the peak seasons, and probably at least several hundred stellar sea lions present during peak season in the study area. These estimates do provide important information that are concerned about sea lion predation on fish stocks, which is obviously a big concern. And we feel the results also may be useful in helping to evaluate potential reasons for the relatively recent increase in numbers of big killer whales in the study area as well. More sea lions might mean more prey for big killer whales, and so there might be something there that might be a possible causative factor or partial causative factor. So just a few words on the status of the project. The report for the Puget Sound partnership was completed in fall of 2022. We submitted a paper to Aquatic Mammals in December of 2022 from that report. The paper is still in review. We haven't received the reviews back quite yet. We submitted our data to the Puget Sound partnership in 2023, April. And currently, as I said, the paper is under review, waiting reviewer comments from Aquatic Mammals. And we are currently prepping a data submittal to OBIS CMAP online database. We are hoping that if we get the reviews back from Aquatic Mammals soon, and that the reviews are favorable, and the paper eventually becomes accepted for publication, we can see it published maybe in mid seems pretty unlikely at this point, I suppose, but maybe late 2023. And we obviously would like to see follow-up studies done on this. We did, as I mentioned, have some challenges and shortcomings from using these data, and we feel like we made some progress in seeing how future work can be done to sort of avoid some of these shortcomings and come up with even better estimates in the future. So thank you for your attention. I'd also like to thank the funding sources for this project. The Puget Sound aerial surveys were funded by the US Navy. The Northern Inland Waters were funded by the National Marine Mammal Laboratory. The sea lion tagging work was funded by the state of Washington, NOAA Fisheries, as well as some other in-kind contributions. I'd like to thank Sharon Mellon and Peter Olesiak primarily for providing tagging data. And again, this analysis itself that we're presenting here today was funded by the Puget Sound Partnership, Salish Sea Marine Survival Project. So thank you for your attention, and I'll leave it at that.
Thanks, Tom. Want to open it up to questions from work group members. You can raise your hand, unmute, put it in chat, whatever you prefer.
No questions? Wow.
I'll raise a hand.
There's got to be some questions. I was gonna say.
Yeah, what do you think is driving the abundance? Is it herring? Is it?
That's a good question. Yeah, I mean, first, if you look at the long term, obviously, the main thing that is driving the abundance, I think, of course, is the recovery of the populations from, human exploitation. But if you look at more shorter term, what's going on, we don't really have any trends data from this, so it's kind of hard to say. Obviously, herring abundance and other prey abundance must be a significant factor. I always wonder, whether or not, as I mentioned in the talk, the situation with big killer whales is also a factor here as well. Mm-hmm. If indeed the data that we found might suggest, if some of the previous sort of guesstimates were accurate, that maybe there are somewhat less sea lions in the study area than had been there in the past. Again, those estimates are not, very precise, but if they are accurate, maybe the number has gone down in some cases a bit, and that might be related to big killer whales increase in abundance. So I would think that those would be the two major factors, but it would really interesting if we could do, another set of surveys and be able to, look at trends over this, 8 to 10 year period from when we did the original surveys.
Yeah, just further comments. One is that DFO has recently done a report on their California sea lion surveys in BC waters, but it's been internally reviewed, but it's not yet available.
Okay.
But it's Strawn Tucker would be the person to reach out to if you wanted to sort of compare notes unofficially.
Yeah.
And I think the other thing interesting is that it appears that California sea lion numbers are stabilizing in terms of pups born. And I would guess that that will over the coming years filter out as they age. And we should see some stabilization in California sea lion adult males in the Salish Sea.
Right. Yeah, makes sense. Definitely. Okay, thanks.
Rob?
Thanks very much, Tom. This is just such a great study. It's neat to see it. I'm a big fan of using line transect surveys and density surface modeling to understand at-sea distribution of pinnipeds. I had two quick questions of clarification. Once you've done that careful filtering, which of course you had to do to censor out the sightings, you get down to relatively few sightings that are, so your overall abundance estimate is really sensitive to the decisions you make. Yeah. And I just wanted to clarify the tagging data that you use as your multiplier for hauled out animals. It was once referred to as animals hauled out, and then in another place it was wet dry. And so I'm wondering if you have 25 or 100 on effort sightings, and that includes some animals that were hauled out on buoys, are you double counting those?
Yeah, that's been probably the most challenging. This is the third project that I've been involved in, attempting to use line transect data that were sort of generally collected for marine mammals in general, and using those to estimate abundance for pinnipeds in a relatively inshore or coastal area. And we've come into the same challenge every time. Is what do you do? Obviously, for cetaceans, it's pretty straightforward. I mean, you're setting your transect lines up over the water. You're estimating animals that are, unless they're stranded, they're in the water, right? But for pinnipeds, you've got this very, very challenging situation that you've got animals pretty much all the time that some are on shore and some number are in the water, and you never really know exactly what that is. And so it's always really challenging. And the biggest part that we found really difficult to deal with is what do you do about those animals that are on land or on some kind of a solid structure, but that are in a situation where it's not clear whether or not they would have been counted in the regular, pinniped colony counts that are done. Obviously, the regular haul out spots and the places where the animals breed are going to be places that are going to be targeted for the aerial survey colony counts. And so, you can kind of avoid over counting or double counting those animals by not including those animals in the estimates that you produce from the line transect work. But the real challenge is these animals that are, for instance, on buoys, or even more challenging is animals that are on sandbars or other things that right now might be dry because they're on a sandbar at relatively low tide, but in 4 hours, that same sandbar is submerged and the sea lion in the same place is now going to be in the water. So what's in the water on land actually can be different in different times in those, shallow areas like that. And so we've kind of come up with this solution that I'm never very satisfied with, but it's sort of like the best we can do at this point by trying to look at it from the perspective of when the aerial surveys are done traditionally, what animals would they likely be seeing? They're not likely going to be seeing animals that are in the water because they're not really surveying and counting over the water. And they're going to be primarily focusing on those haul outs or colony areas. And so we figured that the animals that were on buoys, because the buoy itself is in the water and the animals on the buoy usually for relatively short periods of time, those animals would be more likely to be associated with animals that are in the water, even though they're not technically in the water when they're on the buoy. So luckily we didn't have a huge number of sightings to deal with like that. So it probably wouldn't have an enormous impact regardless of how we handled it. But it's always been the most frustrating part of trying to do this kind of an analysis is how do we deal with those situations? And with the harbor seal study, we had an even more challenging thing because we did have a lot of animals on sandbars and mud banks that were, maybe their bellies were wet, but their backs were wet. So it wasn't even clear whether the actual animal at that particular moment was wet or dry or in the water or not in the water. At least the sea lions, we didn't have many of those situations to deal with. So it was a little bit more straightforward for these guys. But yeah, it's a challenge, and it's something that I think it would be really useful to see some directed research done at looking at how, whether it involves computer modeling or specific studies, using multiple platforms or whatever. Some work I think really could be done to look at how we can better address this in future studies like this.
Yeah, and I don't mean to belabor the point, but I think one of the strengths of what you've done is that because the line transit surveys, the aerial surveys, average over tidal cycles and so much time of day, so much of the variability that goes into that multiplier.
Right.
And the fact that you counted in your aerial surveys seals or sea lions that were on land, you actually have an opportunity, of course, to use to generate an abundance estimate for the hauled out sea lions you detected on your line transit surveys. And it would be really interesting to see if the ratio of animals that were on land to on water. I'll send you a paper that Len Thomas helped me do in BC. This was mostly on harbor seals, not sea lions, but
right.
There's information contained in the ratio of your sightings on land versus on water, in water. And ideally those proportions would be identical. And so you're effectively ground truthing the correction factor that came from tags, wet, dry tag studies, all out, in water studies from other sites, other times of years. So I can send you that paper if you think it'd be useful.
Yeah, no, it sounds like it'd be really useful to look at. And yeah, that's a very good point. We didn't take that approach in this study, but it's something that we could certainly go back and take a look at. The data are all there, so we could certainly go back and take a look at that retroactively. So yeah, that's a good point, definitely. Thanks.
And we'll do one more question, Alejandro. And any additional questions, Tom, if you're able to stick around.
Yeah, I heard
See you again. Well, echoing what people have said, great study. Thank you for taking all this historical data and putting numbers for folks. A quick question: do you have a sense? Can you model how sensitive your estimates are to the assumption of no seasonal variation with the tagging that? Is there a way to have a sense for how robust the model is to those variations or not?
I'm not the modeling guy. Eric was our modeling guy, so it's unfortunate that he's not with us today, because he would be able to answer this question a lot better than I would probably. My feeling is that probably with the data that we have available right now, we really couldn't do a very adequate job of doing that. I think what it would require, from my perspective, probably is further tagging work done during some of those other seasons, so we could get an idea as to whether or not the assumptions that we made in this study were in fact, justifiable. In other words, getting actual data on what the relative portions of animals in the water and on land are during those other seasons where we didn't have tagging data from, available for this study. But as far as whether we could do it from, kind of like looking at the existing data, I kind of don't think we could, but again, it's a modeler question, I guess. I'm not the modeler guy.
Yeah, no, I can always shoot Eric an email. But brings me up to my second question. You mentioned follow-up. Maybe I missed that part. What would that entail?
Sorry, could you repeat it again? I missed that.
Your follow-up study that you mentioned, because you're right. I mean, it's data from 10 years and there's some shortcomings with the gaps, right? So what's the follow-up that is being planned?
Well, there's nothing really planned at this point. It's just sort of, recommendation, suggestion, and that kind of thing. But obviously, if we had, a new set of surveys, done now or in the future, we'd have the ability potentially to look at trends from that 2013 to 2016 study period to the new study period and see whether or not, the estimates for particular seasons in particular areas have gone up or gone down. And also, I think in our paper submitted to Aquatic Mammals, we do recommend some specific tagging work done to try to address some of those assumptions that we had to make. Again, doing some tagging work. The tagging work that was done previously always had specific, goals in mind. And that's why the animals were tagged during particular seasons in certain areas. And of course, we've then going back and using those data for a different purpose. So they didn't really fulfill the needs that we had for them very well. But if some in future tagging work actually were to look at that same information during those other seasons where we lack data, that would give us a ability to sort of go back and see whether or not those assumptions were justified.
And we're gonna, thanks so much, Tom. We have a question in chat as well.
Thanks, Tom.
If you could answer it in chat, that would be wonderful, so we can.
Sure. Yeah, no problem.
Great. Thank you. Our next presenter is Hendrik Nollens, presenting on the Southern Resident Killer Whale remote health metrics. And you might have more of an introduction, Hendrik, but you should be able to share your screen and take it away.
All right, that sounds good. So I'm Hendrik Nollens. I'm a veterinarian with San Diego Zoo Wildlife Alliance. And for those of you who don't know me, here I oversee the hospital, pathology, diagnostic, and disease investigation teams in our work here in San Diego, which is where I'm based, but then also in the field programs. I'm new to this meeting. Usually it's, I think, Joe Gatos attends, our partner in crime. Joe attends this meeting. And so to those of you who are hoping for 15 minutes of entertainment by Joe, I have to disappoint you. It's just me today. Yeah, but so yeah, I was asked here to I think introduce and to some of you maybe update on our initiative to develop remotely collected health metrics for southern resident killer whales. I'm going to try to share my screen here. So because this is my first time attending this meeting, I didn't quite know who my audience was going to be. So I hope you'll forgive me for any inadvertent indiscretions. I can still see you and I can see my desktop. Do you see the slide? Do you see a title slide? Okay, great.
We can see it.
Okay, great. All right, now I got to move my clicker over here. Here we go. All right, so as far as introductions to the project go, the initial team for the project, the idea for this project was really all conceived by Brad, who I see is on the call, which is fantastic, and Joe. And so pretty quickly, the initial project team consisted of Joe, Brad, they brought in myself, Linda Rhodes, and then obviously Brad's team, including Maya, who's also on this team. The project team has this started in 2018, 2019, at least the field component of it, and our team has been growing, which is fantastic. We have expanding our collaborations. One thing I sort of wanted to point out is that what I think is unique is that the project is a partnership between biologists, conservation managers, and veterinarians. So there's sort of some unique perspectives in looking at some of these problems that you all are looking at. And it's between government and the non-profit groups, which you are familiar with. But all the work is we try to exclusively do based on working with teams and boat platforms that are already on the water. So not adding any additional burden. The vision, the concept really came from Brad, but that got kicked off by some killer whale health focused workshops up in Seattle, I think 2 or 3. I forgot what year those were, but then the concept of health and needing to understand health really got accelerated by the gaps in knowledge that were identified in 2018 as part of the veterinary intervention or response to J50 at that time. I'm excited. I hope this doesn't bore you, but I'm excited that the Puget Sound Partnership uses conservation standards and Marathi for your conservation planning because we do too, and that means we speak the same language. So in the next few slides, and I'll do this real quick, this is also very high level. I'm going to show you how we are capturing the situation that we're trying to influence with our work. So this is a situation model, very much from a whale health-centric point of view. So not from the Puget Sound point of view, but really looking at the situation from the whale's perspective. On the left here, the pink boxes, you'll see the human activities that are leading to ecosystem level threats. You know what they are: the river dams, the indiscriminate fishing, etc. I don't need to explain that for this group. These threats have been historically pretty well identified, and mitigation is attempted, right, to varying degrees. And in the green boxes, then from left to right, you see the experiences that that these human activities are triggering onto the on the whales. Reduced prey, interference with foraging, bioaccumulation of pollutants, prey shifts, and they go through this node from the whale health centric perspective of altered health, which can then goes to decreased survival or decreased recruitment. Ultimately, from a sort of a veterinary point of view, looking at this, what the whales are experiencing, the stressors directly onto the whales are altered age structure, altered health, decreased body condition, inbreeding depression recently reported, reduced Chinook availability and reduced population size, which then is, of course, these stressors then lead to decline of our conservation targets of southern resident orcas. In more recent years, I believe with several of you, the correlation between these historically recognized ecosystem threats and the SRO decline has become less tight, and especially forward-looking. So there have been some questions raised about climate threats and species threats that maybe we should consider. Climate change is an existing threat amplifier, but it also creates some potential new impacts, such as increased or bigger harmful algal blooms in the area, as far north as Alaska. And in addition, our project team is asking if pathogens could be an obstacle, even if not in the actual decline of the species, but potentially in the hurdle in recovery with all these mitigation measures already in place. Regardless of which pathway is real or hypothetical, and the weight of each of these pathways, they come through this altered health node, which then leads to affecting our conservation target, southern residents. And so this is then this altered health node is where we target our intervention using conservation standards terminology, southern resident orca determinants of health. The objectives of this project or strategy or intervention are 3 or 4-fold or 5-fold, depending on how you slit, if you're a lumper or a splitter. The objectives is really to work towards having to expand, because some existing work is going on, local year-round capacity for health evaluations for southern resident killer whales, to use that data in relatively real time to aid in the determination of what is a healthy whale versus a non-healthy whale or vulnerable whale, depending on which term you choose to use. Hopefully help inform management decisions in more real time, but importantly to also explore or confirm some of these existing or proposed pathways that are leading to lack of recovery or originally the population decline. There is a fifth objective that, obviously with J50 in mind, is that any information we glean, we learn, will actually help inform future interventions of whales of high conservation values, such as young females with their entire reproductive life ahead of them. So putting on Joe and I putting on our veterinary hats here. Traditionally, a veterinary exam consists of a few components. There's sort of a template for that is very universal across species. One is signalment, who is it, background, what else is going on. And then there are a set of subjective or qualitative measures that you look at that are just sort of a judgment, such as things like, how does the animal look? How is it acting? Is it slow? Is it, you name it, subjective assessments. And then there are a set of objective measures you can take. In some species, it could be a weight, size at age, growth since last saw the animal, but a number of them are listed here. So from the traditional veterinary protocols, also from whales that are cared for and that are in human care, and health assessment of other wild cetaceans, usually smaller ones, we developed a list of 52 potentially useful remotely assessed health metrics for Southern resident orcas. And these can be derived sometimes just from records, family histories, but also boat-based observations, boat-based sample collection, drone-based observations, and drone-based sample collections. And for the past 4 years, we had a bit of a break because of COVID, of course, we've been evaluating primarily focusing on the feasibility and the usefulness of each of these 52 original metrics. And I will run you through some of these. Visual qualitative. The Southern residents have a tremendous asset, tremendous advantage that there's a very experienced biologist team on water with them a lot of time. And these individuals can routinely identify individuals and give us qualitative data that is collected visually on animals. This is social grouping, interaction, social speed, swim speed, activities, what seems normal, what seems abnormal. And Dr. Giles from Wild Orca is working actually on a tablet-based app that can be used by several teams to upload that data to collect the data in standardized ways and upload it to the cloud at the end of the day. We also have some visually collected quantitative metrics like skin condition, respiratory rate, and see Rob's on this call here, which is great. We're working on getting and seeing if we can get normal values using theodolite collected swim speeds and associated with respiratory rates, so we can get respiratory rates at speed. As you well know, we mentioned drones earlier. Other groups, John and Holly, are doing body condition scoring, which is a tremendously valuable parameter in the assessment of a health. And then we are collecting fecal samples. Here you see Brad at work. We collect fecal samples from the animals. We are able to genotype them in-house back to the individual, so we know which whale the sample's coming from. And then we use the rest of the sample for a number of quantitative tests. Cytology, gram stain, parasitology, aerobic culture, hormone analyses, not done by us, but again by Wild Orca, and now more recently also looking at harmful algal toxins. Brad's pretty good at fecal collection and finding those things, but even better seems to be his little EVA. Everybody knows EVA, I'm sure, on this call. And then, of course, I had to put in this photo of Joe. How could I not, since he's not here? Breath samples. The respiratory tract is a very commonly compromised organ system in cetaceans. When a cetacean is sick, a lot of times you are dealing with a respiratory infection, either primary or secondary. And so therefore, breath and respiratory tract breath samples are a key veterinary data opportunity, but also particularly challenging in this species. The way we have been going about it is that our conservation technology team conducted this computational fluid dynamic model and designed a UAV with a collection plate that can be placed in certain parts of the drone. It's reusable, so you can have a different plate for every mission. There's an option to actually include suction onto the device with a little servo. And the technique is to focal follow the whales and then drop in after the animal has exhaled and dropped below the surface. You've probably seen these images or similar images from other species before. This makes it look easy, but I don't fly the drone, but it really is not that easy at all. So sneaking up on following the whale exhales, submerges, and next thing the drone comes in, and you can see sort of from that angel wing signature in the plume that we actually have hit some of these droplets and collected them onto the plates. Again, we take those home and perform a number of lab analyses on those droplets. We also tested the usefulness of infrared and imaging and combined infrared and actually exhaled CO2 imaging. And the purpose here is, sorry, this has some animation in it. Let me skip through that real quick. Yeah, the purpose here is we're really trying to get at developing respiratory health and respiratory function metrics, such as tidal volume, the amount of air that's being displaced when an animal exhales. As you can imagine, there's a lot of drivers of variation in that. So this is actually a very challenging one for us, but it's such an important one that we continue to work on ways that we can estimate this tidal volume whether it's through plume height or ejection rates. More to come here. The infrared imager also allows us to identify hotspots. And so, for example, this is what the world looks like when you look through one of these cameras, and you can see these areas that are warmer, green, yellow, than the surrounding part of the whale, the blue or the green. This is normal to some extent in cetacean. They have thermal windows. They regulate how they shed how much heat. But the question is here, one of the questions that we're in the process of answering is, can we use this infrared to detect whether skin lesions or trauma are active or recent versus chronic changes? We can't take the stethoscope onto the water. So we are experimenting with a directional microphone to collect breath sounds, to listen to the respirations, the expiratory part of the cycle and the inspiratory part of the cycle. We are navigating and exploring ways to do this in a standardized way, collecting the sounds, the soundtracks, I guess, if you wish, but also then we look at both subjective, as you might as a clinician, but a standardized automated analysis for some of these sound signatures that you can derive from the audiogram. And then most recently, this past season, we actually brought in we looked at a new platform. We had a larger drone that fly at higher altitude that allows for higher resolution imagery. Obviously, these are not residents in here. And so one of the questions we're evaluating, again, with other project partners is, can we use this platform or such platforms or partner platforms for detecting or could be significant observations in health evaluations, such as interest in food or defecation rates. Most recently, and this is literally, I want to say a month ago, but it looks like it's actually 5 or so weeks ago. About 5 weeks ago, we convened in Seattle. We hosted a workshop with conservation biologists, veterinarians, resource managers. The list of attendees is on the left here in the slide. Several of you were invited. Some of you were able to attend. And so really, this was a report out on, a 2-day report out on what I just presented in 12 minutes or 14 minutes. But in greater detail, go through each of these metrics. What have we done? What have we learned? How successful have we been? What sample have we gotten? What data have we gotten? And then with this group, really talk through, what does it mean? Is this potentially something useful? Or, is the spread of whales completely not correlated to how much food is available, et cetera? So not just veterinary data points, but a lot of these observational behavioral data points as well were reviewed. The output of this, obviously we are not ready with this. This is just 5 weeks ago, the plan is to put together a NOAA technical memo. And for each of the 52 health metric categories, health metrics that we started with will be assigned to categories. Some that are ready to use and implement, ready to use and interpret. Some a prioritized list of metrics that have remotely collected metrics that have potential but need R&D, need work, but technical development. There's bound to be a list of research needs, getting data and looking for trends to really help understand and inform what is normal for a southern resident versus what is abnormal. Some of the metrics we are moving into the parking lot because it's too difficult, unable to get quality data, not reliable, too much effort. You could be doing 10 other things instead of just one thing. And also, we also did a bit of horizon scan. What other tools are coming in wildlife medicine that we may be able to modify to use from one of our platforms. Then for us, next steps is this summer is to now actually go through all this data and decide what are we going to focus on this summer. Do we start collecting data, or do we focus on optimizing some of these techniques that are not yet ready to use and interpret by the on-water teams? I have to acknowledge funding agencies, of course, several. Thank you to all. And I think that actually concludes, that's my last slide. I think it did better on timing than I thought I would. But happy to take any questions.
Thanks, Hendrik. We do have a question in the chat from Scott. Scott, did you want to share that out? We'll also please raise your hand and for any questions for Hendrik.
Yeah, sure. Thanks, Hendrik. That's very interesting to see so much innovation. I guess I have two questions. One I put in the chat was just really an idea that the spatial data, either from acoustic detections or visual detections, is definitely increasing in the last 5 years, thanks in part to social networks, Facebook, but also people getting better and better cameras. And it's not uncommon now for land-based photographers to get an individual ID on a southern resident as it moves across the region. So I just wanted to flag that, like, there's more and more data sources and they're getting better and better. And so for individuals, I'm wondering if poor body condition might be correlated with some threshold in travel speed, let's say, between two long distance, like widely separated points. So I just wanted to flag that. I'm not sure that anybody's done that, but we have been starting to talk about, oh, they're coming south in Arrow Strait. How long will it be before we hear them at Port Townsend? So we're already doing some inference based on, published information about travel speeds, but it's the first time I've thought of it as a potential health metric.
Yeah, there are people on the screen who know far better about what would be normal, what would be abnormal for travel speed for one whale to take longer than the rest. It is getting at something that we also are getting at, which is this individual animals that are lagging unusually. And the way I think of it, or have thought of it until your question just now, is and the way the project team has thought about it is, where is it relative sort of the pod to the whales, the animals traveling with? Is it lagging? Is it dragging? But maybe there's another way to go about this. I don't know if Brad or Maya or anybody else wants to chime in. We do know that decreased body condition changes buoyancy, swim patterns change. You get a little bit more of that, more of a bow wave because they're working more to get through the water. Then you burn more energy, so it's a vicious cycle, right? But it could be one worth looking into. It's currently not on our list. Andrew, sorry.
Yeah, hi. I'm impressed with the full list of everything, but I'm wondering, where does inbreeding fit into your health assessment? And is there a way to score each individual so we can add that as the added risk, which complicates all the other threats?
So I am not a conservation geneticist. I'm just a user of the data. It is my understanding that from the data, at least from looking at that paper that was published a month ago or 2 months ago, that there's a inbreeding score that is assigned to each whale. And you can put that in. You can include that in your medical record, in your veterinary record, if you wish, for that animal. That's a consideration. Generally here at San Diego Zoo Wildlife Alliance, when we've had recovery programs that recover species from really low numbers, there's inbreeding, right? You can't, you just allow it because you can't circumvent it. How do you go back from 30 individuals to several hundred without going through a genetic bottleneck? The inbreeding itself, when I talking to my colleagues here, is considered like climate change, really, is sort of a threat amplifier. It makes it even harder on the animals, or you have to assume that it is, if you assume that the amount of inbreeding is actually making them less fit, then it just makes it harder on the animals. So you actually have to improve the circumstances even more to allow them, to give them a chance to recover and expand. But there's an assumption in there. Not all breeding immediately leads to impacts on fitness. It actually has truly physiological impacts. And but if there are, we don't have for a bird program or a small rodent recovery program, you can bring in other individuals. You can translocate. You can bring in founders from other populations. Clearly, that's not an option here. So really, what we can do is improve the support infrastructure that's available to the animals.
It seems to me that inbreeding is the elephant in the room, but I don't see it being explicitly addressed. I'm just wondering, are you going to add this to your list and into the flow chart of threats?
It was, maybe I went too fast, but it is captured as in the situation model. It is one of the 6 or 7 stressors is inbreeding. It is there, right? We can't ignore it. But the interesting work would be to do the work we've been talking about with Brad and some others, is to look at, okay, there's this, but what is the impact on fitness, and see if there's actually, what's the practical implication of the degree of inbreeding that has been documented now.
Thanks.
But it is, Andrew, it is on the model. It belongs on there. It's a stressor that the whales are experiencing.
Thanks, Hendrik. Are there any other questions? We're gonna take a 5-minute break instead of 10, just to make up some time. But happy to, if anyone has questions, put in chat. Maybe Hendrik can take a stab at answering any that pop up. But let's plan to meet back here in 5 minutes. So we'll return at 1:02. Oh, sorry. I was rounding completely wrong. We'll return at 2:32. So take a bathroom break, get some water. I'll put this in chat. And thank you, Hendrik. All right, we'll be filtering back. You don't have to turn on your video, but you can put a thumbs up if you're with us, so we know we can get started here. Let's give it another minute. Still 2:32, so. Right, so thanks all for accommodating with the shorter break. And thanks to both of our presenters, Tom and Hendrik. That was great. There's another question for you, Hendrik, if you're still, yes, you're still with us, that Scott posed in the chat, so feel free to answer in there. We're going to take a little bit of time today just to review the 2023-24 work plan update. This is something the group briefly touched on back in December. We did a mural exercise for those who attended that December meeting, where we talked about which objectives and goals should be carried onto the next year work plan and what new work plan elements should be considered in this upcoming year. This work plan will it takes place like July 2023 to the end of June 2024. So keep that in mind as we review it. What we're going to do is just quickly walk through the objectives that have been drafted by the work group coordinators with Scott's assistance and input as well. Run through it and just ask if there's anything you don't see represented in the objectives that you think should be there. And then, as I kind of previewed when we were discussing the vital sign, we'll take a similar approach where we'll be sharing the work plan through the listserv, asking for comments and feedback, edits before we finalize. The work plan's deadline, or is like due the draft by the end of June. So my plan is to get this out to folks in the next week or two. And then we'll work on finalizing, integrating the edits, and then we'll have our final work plan for 2023-2024 in July. Okay, I'm going to share my screen, again, walk through really briefly, and then return the conversation to the group of if there's anything you think is missing, anything you think we should add. So I'll run through the objectives here. And again, a lot of this is carried over from last year's. So one would be to promote information sharing and collaboration among work group members. Then in the next column, we have the specific tasks as that we would take to achieve that goal or objective. So again, kind of staying with the typical cadence of 3 work group meetings per year. Potentially, we talked in our last meeting about holding an in-person as an option for the fall or maybe in 2024. And then you'll just see a list of tasks that are for achieving that objective. 2, we have the vital sign reporting. So updating the ORCA sign key messages. And these we revise every 2 years. We just went through the revision process. So this isn't something we'll have to do next year, but keeping it up to date that this is something we'll be carrying through the work plan. And of course, if there are things we want to revise, we can do that annually, I believe. It just wouldn't be carried through to the state of the sound report, which is published every 2 years. And then the ORCA's vital sign updates. So that's updated annually. Lynn Berry is the vital sign indicator reporter for the number of southern resident killer whales. So that's something we'll have in our work plan. Increase connections and collaboration across work groups. This was something that was flagged as an interest to do in the upcoming work plan. And we have some task breakdowns here, calling out specific work groups and topics of interest in collaboration. 5, support work group member activities that inform Puget Sound health. So this would be, we're going to be talking about it a little right after this agenda item, but submitting things to the 2022 Marine Waters Report. It's an annual submission, creating profiles on different marine mammal species. 6, leverage the new orca occupancy indicator for other species and objectives. Again, this was something that was flagged as a objective that we should consider in next year's work plan. 7, support work group members seeking opportunities for learning and sharing at relevant conferences and our speaker series. We know this is, we'll be seeking input specifically from work group members of what those conferences or speaking opportunities include. So that will be a specific ask for folks. 8, continue information sharing on orca vital sign ancillary measures. So those include body condition and fecundity of southern resident killer whales. 9, make stronger tribal connections. This was also a recommendation at the December meeting. 10, conduct an orca recovery monitoring inventory and gap assessment. This is something the work group coordinators would lead, but a subtask of that would be presenting it to the group to get feedback on. So those are our 10 objectives right now. I know it's a lot to take in, and it might be easier to provide feedback when you have it in front of you and you're able to read through everything. But just want to pause here and ask if anyone has any initial feedback, anything they think is missing or you'd want to discuss in this setting right now. Just going to stop sharing my screen so I can see everyone. And I don't see any hands, so I just want to review the next steps again. That this table is just one component of the work plan. There's going to also be the vision, the more narrative goals, and the year in review as well. So that's the document that will be shared out. Again, I'll make specific asks in the email, but it'll be a similar process that we went through with the vital signs. So looking for any feedback that you have once it's shared with you all. And if there's no questions or comments, we'll go on to the next agenda item, which is the Marine Mammals Work Group Species Committee walkthrough that Scott will be leading.
Great, so maybe share my screen now, if that's all right, Nicole. Mm-hmm. Nice work getting us back on time, by the way. Oh, it's disabled screen sharing.
Oh, sorry, Scott. Let me make you a co-host.
No worries. One thing I just realized as you were going through it is to flag for folks is that the partnership is leading along with the Puget Sound Ecosystem Monitoring Programs Steering Committee, an effort to rethink and re-envision what a PSEMP website might look like, as perhaps something more than what it currently is, which is a set of box notes within the Puget Sound Partnership website. So that's to the first point, as a tool potentially for us doing a much better job of sharing and collaborating, especially between meetings. Like that is a coming innovation that I'm excited about. I don't know what form it'll take, but I think it'll be much more advanced than what we currently have. All right, so trying to share now. All right, you seeing my screen okay?
Yep.
All right,
yeah.
So I think most of you have seen this before, but I wanted to just take the time to explain how I've been using this spreadsheet. It basically shows the committees which we've formed over the last 5 years. and the members who volunteered to focus on particular species or topics or ecotypes. So mostly we've got species here, but there are the 2 ecotypes of killer whales. And we did form an acoustics committee in the last year. And there's also, you'll see, an ancillary measures committee, which we haven't formed, but we might want to with respect to the current work plan. And it might tie into some of the things that you discussed, Hendrik. Those ancillary measures currently are, I think, all related to Southern residents. So it's possible we can just bundle that into the Southern Resident Committee. But that's that column. And then this is an opportunity for folks to add themselves or subtract themselves. Usually once a year, I try to cue folks to think about whether they'd like to change their commitment levels, or if they know any new colleagues who might enjoy participating in committee activities. We haven't used these much, but I'm trying to prepare for the PSEMP website and the ability to provide more timely updates than what we've currently been doing. And more data products from the work group in general over time. There's also columns here for lead authors, proofreaders, and prospective authors. If you think of someone, feel free to add it here and I can reach out to them. And then we have communication channels. So these were triggered by the new strategic plan for PSEMP, and that effort to try to think about how do we communicate what we know. Traditionally, the main way we've done that is through the vital signs. So this column is historically been the main focus, is ensuring that the vital signs get updated annually, or the key messages get updated every 2 years. So that's green. My goal over time, and I think the goal that I'll take forward with any new leadership in the next few years, is to try to keep more of this green for all species and topics as we go forward. So a couple of these other channels that are established and ways that we can provide updates to PSEMP, the PSEMP community and other end users, are this Marine Waters report that the Marine Waters work group puts out annually. It's been traditionally a hard copy report, and we've contributed to it quite a bit over the last say 5 to 10 years. And I think especially with the new website, and probably as soon as next year, it'll become a digital only publication to be more cost effective and maybe faster. And then there's the Encyclopedia of Puget Sound, which if you Google Puget Sound and your favorite marine mammal species is typically what comes up first in a Google search. So this is a place where we've been trying to add content over time with some stimulation from Jeff Rice, managing editor there. So you can see a number of these yellow contributions have been made with leads from folks like Cindy Elliser and Monica Wieland in the last couple of bienniums. So those are sort of the axes. There are links here to the existing content that we have in the Encyclopedia of Puget Sound for those of you who would like to see sort of what we've done so far. And then there are listservs that I've set up for each committee, but we haven't used very much. But those were meant to be for more granular communication than if the whole work group. And lastly, one thing new that I wanted to share, and I shared at the Marine Waters report workshop, is thanks to many of you, we have a shared album, which I'll just show you briefly here. I'm hoping to get a few more contributions from members, but it's grown quite a bit over the last year. And the idea here was to prepare and agree upon like what the attribution should be, with pretty minimal constraints for PSEMP or Puget Sound partnership use. So the folks who have contributed are willing to have the partnership or PSEMP use any of these images for, for their work. So there's quite a few Southern resident photos here. I think that's the most number we have. It's quite a few Biggs killer whale observations and photographs. These are examples of ones taken from land or sea recently of Biggs killer whales. And we've been trying to get more showing predation events on different species. This was one that Ashley Whitman took of a seal getting eaten. Humpbacks, seals, and John Scordino contributed some showing seals eating things other than adult or juvenile salmons. We've got a few minkies, but there's many more, I'm sure, in all of these, and so on. So I invite you to, if you need some eye candy or you need an image for one of your products, that's a growing resource. All right, so that's kind of the overview. What I guess we were hoping to do, and stop me if you want me to go a different direction, Nicole, is basically work our way through these in the next half hour and talk about potential updates, maybe modifications of our work plan if we discover that we need to do more work on any particular species. Does that sound like a reasonable way to proceed?
Yeah, and I know we had flagged from a couple folks that might have verbal report outs related to several of these subspecies. So maybe we'll start with those.
Okay, yeah, that's a great idea. Oh, I forgot, before we do that, one other thing that I've been finding quite useful, just myself, and it might be useful as we start to have deeper interactions within these committees, is each of these species names is linked to a folder where all the folks who are in the working group committee should have right access. So one of the things that you'll see in there are drafts of short or long reports about the species, but also I use this quite a lot, this sort of the notes about killer whales or any other species. So I try to take, when it comes time to report like what happened in 2022, I find it quite useful to have a list of observations that are hard to remember a year afterwards. So I just wanted to flag that as those, I find those to be quite useful later, and there may be yet another way that we can communicate a bit more about, you know, as between meetings, basically, those notes Google Docs. So yeah, can you tell us who should go first, Nicole?
Yeah, so we can start with the harbor seal. And Cindy, I know I had reached out to you to share any updates, but also welcome anyone in the committee members to share any updates as well.
Yeah, I mean, I could talk just briefly about the WildMe. If I can share, I can do I did a little screenshot thing last night.
Yeah, thanks.
Not that it's we're still in the very early stages of it, but let's
You
see.
should be able to share.
Okay, there we go. PowerPoint. If I can just find it, there it is. Very slideshow. I'm beginning. Okay. So okay, give subtitles a try. I don't want to do that. Why?
And we're seeing your presenter view.
Okay, there we go. Oh, that's weird. Why did it do that? Oh, you know what? Gosh darn it. It's because of the way my monitors are set up.
Okay.
I'm actually just going to turn my video off like that, and then it should be fixed if I do that. So you just can't see my pretty face for a minute. Oh, that didn't fix it, did it?
No.
There we go. Is that better?
Okay. Yep.
Okay. All right. Let me go back to the beginning. Okay. So with the WildMe, they have it's called the Seal Codex, and that's what we're trying to use their seal spotter, the spotting pattern AI algorithm that they have for various different species, but they're testing it out for seals. So the idea, I believe, behind it is the seal codex is actually a seal recognition, so it can identify whether it's a gray seal or harbor seal or something like that. And what we're testing out is if the algorithm that they have set for this with the hot spotter for the spot patterns will work for the harbor seals in the water. And if not, then we have to look about training the AI program a little bit better with more pictures and stuff like that. So this is kind of the testing zone of if this will work as is, or if we need to invest a little bit more. So just again, we're in the real early stages of it because we had to go back and kind of reset how our data was set up to be able to do this correctly. And I'm still learning the codex amongst all the other things that we all do every day. So, but this is kind of what it looks like, the interface when you first start. You can basically, put a sighting in, you can look through individuals, you can look through sightings. But what you would do is you go up to this top thing with a plus and you would say that's going to say, I want to add a sighting. So you would then upload your photo from that sighting and it says, upload complete. This ones are ones that we've already identified from doing it by eye. So then when it goes through, so you do that and you go to the next screen, and then you're putting in all the metadata for it. So how much of the date and they go down to even hour and minute if you want to have real precise timing of everything. And then what detector model you're using, which is going to be the seal one for the seal codex. The region, we did have to ask them to put a specific region in here because they only had a drop down. So there's, I believe, Washington, Anacortes, and then Sailor Sea. There's a couple different ones you can use. And then you can put a freeform location in of a specific one and just more data that you can attach to the photo. So then you go and it says once you click the next button that says, continue, it says the sighting is pending. It's going to annotate. So it's going to put little boxes around saying I identified a seal in this picture versus something else. And so you can't do anything until it kind of goes through that process. And so then next it'll show you that you're ready to commit. So if you're ready to press that button and really commit to your sighting, you can do so. And when you hit the commit button, then it comes up and it shows you the annotations that it have for that animal. So putting the box around, this is where I identified the seal in particular, and then you can see all the metadata that you've put in previously that relates to this animal. So right now, what we've done then is, so you can then run an identification job. So IDing it as a seal. If you go to the custom settings, you can use the actual seal, the hot spotter thing, which I'm still haven't got quite working quite yet, but but manually, at least right now, you can go in and you can create a new individual. So you say, okay, this one's been annotated, it's got all this information. You can identify, you can either manually assign to an animal you already have, or you can create a new individual. So I created a new individual, named it paw print because it's got a paw print on its side. And then there's other information that you can add to it if it has a relationship with some other animal in your thing, whatnot. And then it has all the photos that you've uploaded and the sightings occur here. So this is what I was talking about before. I tried uploading a second picture of an animal I know. And then if you use the custom settings, that's where you can choose to use the hotspot or pattern matcher, which I believe that's what we're trying to see if will work for these photos. And then I hit commit sighting, but it didn't quite work right yet. So I'm just trying to figure out what happened and what I'm missing, because it's probably user error. But ideally that would be the case where you would do this and then it would run through your database and see if it matches with another seal you already have. But regardless, you could then search for an individual if okay, I know this is paw print. So I search for it, it pulls up and says, okay, here's paw print from this date. That other top one's an old one that I deleted. I don't know why it's still there. But then you can just assign that animal to that. And then the next one comes up and as you can see now it has the 2 sightings for that animal. So even regardless of without the AI program working exactly right now, it is seeming like a great place that if you can get the data in there, you can easily pull, it's a whole database, right? So it's not just the matching, it has all the information for all the sightings for all the individuals, and you can cross match and, you know, have relationships with different seals and whatnot. And then on top of that, you can collaborate with others. So I have, I know Alejandro has one, and Scott has one. So we should be able to, in the future, be able to compare between databases and photos to see if we have matches, and that should hopefully facilitate the ability to us to collaborate and work together on seeing where these seals go. So that's my rough and ready presentation on where we're at. And like I said, it's still in the beginning stages, but it's I'm very hopeful, and I'm hoping that we'll be able to do this for more species as well. We're working on one with harbor porpoises as well, but that's a whole nother thing.
Yeah, thanks so much, Cindy. That was the highlight for me when we did the breakout rooms, is just realizing that there's all these new platforms for training AI or applying a model from a different region. Is really exploding, and super exciting to explore the potential for harbor seal individual IDs. Just for context, like these groups, which are WildMe is based down in Portland, and they have now enumerated and identified most of the whale sharks, and working on manta rays and zebras and other things like that. So it's encourage you to explore the platform for your particular species of interest, because they're increasingly open and free and powerful. I think there was a recent paper out about, maybe John can speak to this, but the happy whale AI for identifying individual humpbacks is getting to the point where they're asymptotically approaching, you know, an estimate of the population, because they're not getting as many matches as they used to. So thank you, Cindy, for that little tour demo.
And speaking of John, I believe he had something to share on gray whales.
Yes, thanks. Can you hear me okay?
Yes.
Okay. Excellent. And I think this came out of, I don't know, I think you mentioned someone had requested an update on the UME status. So I was going to present something on that, but I thought maybe more importantly, along with that, sort of link the overall UME to what's going on in Washington State specifically. So that's why I've given it this title. We have a number, Jesse Huggins is our stranding coordinator that's been involved in the responses to the unusual mortality event for gray whale strandings that have been occurring since 2019. But I'll be linking this to some of our other gray whale work and showing you how it connects with things with gray whales in Washington State. And first of all, I would just point out that, you know, when I think of gray whales in Washington State, there's sort of four categories, if you will, that are overlapping boxes. The overall Eastern North Pacific gray whale population, which is what the unusual mortality event is declared for. This is the larger population that migrates, you know, between breeding lagoons in Mexico and the Arctic, these areas shown in red, and migrates between these routes, and twice a year goes by Washington State. There's what we call the Pacific Coast feeding group of gray whales, which feeds from spring through fall in this area highlighted in green from Northern California. And this is what a lot of our research has focused on. This is a much smaller group, only represents about 1% of the overall Eastern North Pacific gray whale population. Canada and some people, including me, think of it as, you know, distinct from the overall Eastern North Pacific gray whale population, because there are some mitochondrial DNA by haplotype proportions that differ between the whales that feed in the Arctic and those in the PCFG, though they both go to Baja to breed, so they would interbreed. There's also a Western North Pacific gray whale population that primarily has been studied in the Sea of Okhotsk, Sakhalin Island, was thought to migrate traditionally down to areas off China and Korea, but satellite tagging and now photo ID matching that we've done of some of those whales show many of them, if not most of them, in fact, were migrating from the West over to the breeding areas in the Eastern North Pacific. So that's raised a little more debate about how to treat that group. You know, is that now just an expanded area of the Eastern North Pacific population? Is there still a remnant Western population that does in fact stay in the West and migrate? And then finally, I'll be relating it to a group of whales we call the Sounders, which clearly are part of the Eastern North Pacific gray whale population, but it's a group of now about 20 whales that break off from the migration in spring, come into Puget Sound and feed, hence their name, the Sounders. And we've been studying those since 1990, and it turns out to be the same individual whales returning year after year. In fact, the original 6 whales we documented in 1990 and 91, all of them had returned for 30 plus years, and it's only recently that 2 of them have stopped showing up, and we think that's because they died after over 30 years. So anyway, just, you know, recap, overall Eastern North Pacific group, that's the UME group we're talking about, Sounders, Pacific Coast Feeding Group, and Western gray whale. So going quickly to the key figures from the unusual mortality event. The current one was declared in 2019. There was a previous one that was documented for 1999, 2000. And as I'll show you, I think there was one previous to that, which was not declared a UME, but there's some indications occurred. And these are the number of strandings by country that were documented. You can see this peak year 2019, when it was first declared, was the largest number of, these are dead animals documented and washed up. It has declined each year. This 2023 number is only partial. And bear in mind that it's probably only on the order of 10 to 20% of the animals that die likely get documented as strandings. So something Rob's, if he's still on, is intimately familiar with, and a topic we've discussed quite a bit and want to report on more, but basically strandings represent just a small portion of the true mortality. And as you'll see, there's some really interesting indications of that being true. These mortalities, and this is just the mortalities in 2019, you know, you can see extend along the entire range of the gray whale, as you see here. You can see most of these mortalities, and these are the ones in US waters, generally occur in March through July. Now, there's some considerations I'll put out there. The Eastern North Pacific gray whale population was at record levels prior to this UME, you know, upwards of 25,000 animals. There have been dramatic changes and decreases in ice cover in the Arctic, but past studies linked lower ice cover with higher gray whale calf production, kind of the reverse of what you might think, because they made certain areas of the Arctic more accessible to gray whales to come in and feed. That previous mortality event of 1999, 2000, gray whales seemed to bounce back from it, as I'll show you in a second. The UME does seem to be winding down. 2022 was lower than 21, and 20 and 19, and 23 is coming out lower than that, but they still have remained above historical norms. So hence the unusual mortality event has remained open. Okay. Now, just to show you the Washington side of the picture, these are, we have a great record in Washington State. I'm trying to put this into a publication because our records go back to the 1970s. And these are the annual numbers of strandings of gray whales just in Washington State. And you can see in Washington State, our main strandings occur in April through June, but every month of the year. And you'll see these clear, oh, I did not, I meant to have I meant to circle them, and you'll see in another slide, but here you see the current UME here, 2019, and then, you know, numbers that decreased a little bit. For Washington State, we had kind of a bigger year than 2022 that occurred throughout the range, but you can see these numbers are quite a bit above what was the normal average of under 5 whales. You'll also see here's that previous UME here, a year right here, and the next highest pair of years, just for reference, and we'll come back to this, 1990 and 91. Just to point out, many of these whales in both UME and non-UME years are emaciated gray whales. In UME years, we see both emaciated gray whales alive and dead. Some of these whales, when they're bloated, they may not look emaciated, but often the blubber layer is very fibrous and dry. lipid levels in the blubber are really low, 5% or less. So some findings there. But there are also other causes of mortality. Some of these may link to debilitated whales sticking around, but you also in the UME years see increased numbers of entanglement, ship strikes, and in recent years in our area, killer whale attacks. Here's the slide I meant to show with those circled periods of highest mortality. What does that link to? I wanted to get to that part. So here's those 3 periods. First of all, here you'll see I've borrowed from a slide of the estimates of the overall gray whale abundance and superimposed it with our annual stranding rates. And I'll just want you to see that in the estimates based on migration counts of gray whales and then adjusted, and this is from work Josh Stewart has most recently done and was reported in a public, a technical memorandum with him and Dave Weller. You can see 3 periods of major declines in the abundances of gray whales based on these counts of animals migrating by. And they line up really well. Here's that 1999-2000 mortality event and a major drop in abundance estimates of gray whales. Here's the count straddling the start of the 2019 UME. And again, and these are big drops. We're talking drops of 6,000-7,000 or more animals, which goes with that theory that the strandings represent a small part of the mortality. And then linking it to our Sounders gray whales, here in shaded bars, you'll see the number of new gray whales joining the Sounders. These are the whales that come into northern Puget Sound, and when have new whales come in to join that? And you'll see by these shaded bars, they exactly line up with these same 3 time periods. Our original Sounders discovered this area in 1990-91, 6 more joined them in 1999-2000, and then we've had 8 more join them here in 2018 through 2021 with this latest UME. So they line up really well. We also have good relationships, significant relationships between the kind of incidental sightings of gray whales in unusual areas like South and Central Puget Sound with those stranding numbers. Sorry, I'm going quick. And I'll just say with these Sounders gray whales, these are these long-term histories. Rows are individual animals, columns are years. Where it's green, we've documented the return of the same gray whale. And here you'll see these first 6 that joined in 1990-91, the next 6 here, and then these ones that have joined in this 2018-2019 period. And there's some other subtleties there, but I'm just going to go quickly with that. Partly because of the UME, we've been working with John Durbin and Holly Fernbaugh to measure the body condition of these Sounders gray whales, both how they are when they arrive and how they gain it. Actually shows, remarkable. This is the same whale in 2 photographs, February then to April. You can see the huge amount of kind of girthiness these whales gain from feeding in this North Puget Sound area. This, I think, is part of the reason why these whales have done so well and survived so well. Though some of the new whales that have joined the group, and this black line shows this one whale that didn't gain, and it's shown here, maybe haven't quite learned. These whales feed in the intertidal zone on ghost shrimp only for a few hours around the high tide. And you have to know what you're doing to get in there and get the full advantage of that. Very high prey densities, but a very high risk foraging strategy doing so in the intertidal in this area with big tides. We have been deploying tags on these whales to look at these feeding habits. This year, for the first time, we used a drone-based tag deployments. First time deploying these on gray whales, working with Ocean Alliance that's been pioneering doing this and has a new publication out on doing this with a number of species. This is a very quick video just showing a drop of a tag on a gray whale from the drone. And you can see the whole perspective of dropping the tag onto the whale and what that looks like. I will point out, just to bring it to our 3rd group of whales, and also mention the Western gray whale, but the PCFG gray whales have not shown those fluctuations in abundance matching the UME, and the Western gray whales have not, as either, really suggesting that the UME is driven by specific interactions and prey-related interactions between that growing population and changing prey availability up in the Arctic feeding area. Okay, there we go. I don't think I did too bad. I know I didn't have too much time, but hopefully that was within acceptable time parameters.
Oh, that was great, John. Thank you so much. And that video was amazing.
A question, John. Real quick one. Do you think they're starting to diversify what they're eating, or you think they're all learning the trick of getting to the ghost shrimp?
As best as we can tell, the latter, but that's the whole reason we wanted to do these additional deployments in the UME years, was to explore that. Is any of that changing? Because I didn't mention, but there's a dramatic increase in the feeding occurring, not just because it's more whales going from 6 to 12 to 20, but we had 2 whales forego the whole southern migration. Actually, I should say 3 whales that ended up doing it. 2 of our long-term whales that showed up in December and early January, and 1 whale that's been there feeding for a year continuously. They're not only more whales feeding, but they're using it for greater time periods. So the share and amount they're taking, the graduate student who Scott, who's been in touch with you, Hannah Clayton, a PhD student at Stanford that'll be working with us on gray whales the next 3 or 4 years, is doing prey sampling and looking at prey densities in some of these beaches where the whales are feeding, to look at how that changes over time and explore that, because clearly their use is changing. And if I can add one other wrinkle, I didn't mention, part of the reason we did the tag deployments by drone, Scott, you will like, and I don't know if I've mentioned this to you before, but part of the goal was to use the drone to be able to tag multiple animals in these closely associated groups. So that little group of 3 animals I showed in that slide, we had all 3 tags on all 3 animals, and all 7 of the drone-based deployments we did ended up covering all members of associated groups. And part of the goal was that we could look at both vocal production, but also use the accelerometer to see if we could use that to identify who the caller is and who's just an animal next to a caller, a key thing for doing things like abundance estimating from vocal detections.
Yeah, I was excited just to hear that they were vocalizing. Yeah, it's just amazing the drone work you're all doing. Thank you. Thanks, John.
Andrew?
I had a question, John, about the ability of ghost shrimp to sustain gray whales, and in particular for pregnant females, since they've got an excessively high energy requirements. Do any of your animals, do you have pregnant females?
Yes, the majority of our sounders that we've sucked have been males, but we do have 2 identified females. And interestingly enough, you see every 3 to 4 years, they don't show up. And those are years we think they have a calf. And now with the drone-based stuff, the pregnancy identification has confirmed that. Our longest term female that we call Earhart, who we think pioneered this feeding strategy, who we now we've been seeing some 34 years in several of the years now before she didn't show up, we documented she was pregnant. In 2 different other years, she was seen with a calf in the Baja Lagoons in the year she didn't show up as a sounder. So I think we were able to link that. So the one thing that I'll point out related to your energy needs question is that even though it's only these 2 animals, I looked at each of the preceding years to when we think they had a calf. And so we either could confirm or suspect they were pregnant. And those animals showed up early in each of those years. And that kind of goes a little bit with what came out of the Dale Rice's early work, that pregnant females may be some of the earlier migrators in the migration, precisely for that reason, because they know they have greater energetic requirements. And it seems like that's reflected with their time up in this area.
So just to be sure I understood you right. So the 2 females, when they had their calves, they didn't come back with their calves, or it's while they were pregnant, they weren't feeding there?
Yes. So they were feeding there when they were pregnant, but they didn't come feed there when they were lactating with their calf.
So you suspect I guess you suspect they went further north?
Well, mothers with calves generally migrate later. And so they're either foregoing coming here because they're already later, and the whales, the Sounders whales feed primarily March through May.
Mhm.
And the pregnant and the females with calves are generally coming through in May. So they're right at the tail end of when they feed there. So they may, we think all of these animals, or almost all of these animals, go on to the Arctic to feed after that springtime period.
Fascinating. Thanks, John.
Any other questions for John? Thanks for that presentation. That was great. I know we also called out sea lions, and we had a presentation from Tom earlier today, but just wanted to throw it back to you, Tom, if there were any other updates coming down the pipeline aside from your paper soon to be published.
The only other thing that I would say is I've talked to Scott a little bit about the idea of doing the Encyclopedia of Puget Sound chapters for sea lions, for the 2 species of sea lions, and just wanted to throw it out there. I know there might be a couple of people who might be interested in co-authoring those, who have some specific information. You know, our information really is primarily on distribution and abundance, but if there's other folks that would like to be involved in that, if you could pop me an email and let me know, we'd be happy to work with you.
Then recognizing there are a lot of other subspecies or species committees, subcommittees. And if there's any other updates from work group members on projects, interest in authoring some content, or if you have funding proposals that you want to flag within this group, opening the floor to members.
While we're waiting, I'll flag that I put in green here the likely contributions that we've been talking with Jeff at the encyclopedia about. So I think the thought was that focus in on an update to the harbor seal profile, which you can go and read. It's quite extensive, authored by Joe Gatos and one of his students, but also now somewhat dated. So that's one priority. And then this, Tom has nicely volunteered to help lead both sea lions. And so I'm certainly interested in helping you, Tom, on the general editing side. And the other thing I wanted to flag is that Marine Waters report contributions can be part of generating these profiles. So I can show you sort of what's been done in the past, but it may be possible to generate a profile or an article for the encyclopedia at the same time we do a 1-page presentation for the 2022 Marine Waters report. And then, I think we've got gray whales as well. Stephanie volunteered to help us sort of provide an update. It really would be a first entry in the encyclopedia. So everything that you provided, John, is great. And maybe we can add some acoustics to that for fun. I know they've been asking for multiple types of media. So that's one thing I think we could start to add to these is some acoustic for the somniferous species, some acoustic recordings.
Yeah, no, I'd be happy, especially I think I'm on a couple of these, but certainly for gray whales and humpback whales, I'm both of those I have intentions and on my plate needing to write up a couple of things related to our work. And I would be happy to work with Stephanie on gray whales and even be potentially down for leading the humpback one and stepping that up. And I will hope to have time to do that in coming months.
Yeah, that would be great. I've flagged that and Minkies for like next year. Maybe we kind of prioritize those. There's a little bit of a treatment that Aaron Gless prepared that we could probably leverage And a little bit of acoustic information was provided by Emily Vierling in this the last report for the Marine Waters Group. So there's some pieces there I think we could help you pull together after you publish what you want to publish in the peer-reviewed literature. Oh, and I should do a shout out also to Stephanie, who did a nice job of quickly adapting when we had the lone beluga. So there's a nice article in the encyclopedia now about the beluga. Guess that's an update.
Thanks, Scott. I think what we're gonna circulate in the meeting summary is just, again, I think a lot of people just had to jump off and this day didn't work for a lot of members, but glad for those who are able to join, but really wanting to update that spreadsheet so we can start communicating through the listserv and through those channels and really ensure that everyone's getting the support they need from the work group coordinators, Scott, and potentially other a co-chair or vice chair that will emerge in the next year or two, so we can help keep track and ensure we're tracking what the Marine Mammal Work Group produces. So that's something that we're going to be flagging in our work plan for the next year. Are there any other updates the folks would want to share out before we move on to future agenda items and just wrapping up here?
I could just mention one thing. I'm sure a lot of people on the group are probably aware of this, but there is a paper that has been submitted for publication to officially propose splitting out resident killer whales and Biggs killer whales as separate species. Of course, it's still, in the early stages. The paper is undergoing review right now, but hopefully that will be accepted. We may, and of course, if the paper gets published, then the next step, as probably most of you know, is that the Marine Mammal Taxonomy Committee would be voting on whether or not they agree with those proposed changes. And if they do, at some point soon after that, it would become official. So after many decades, of course, of a lot of people talking about this, there's starting to be a specific plan of action to actually make it happen. So I'm involved in the project in a relatively small way, but I can keep people apprised of the progress of that as it moves forward. Pretty exciting.
Thanks,
Sounds
Tom.
exciting.
Yeah, and the work group typically doesn't meet in the summer, so we would convene in the fall later this year. So perhaps there would be a major update to integrate into that meeting.
Yeah, hopefully the reviews will go pretty quickly. They've been taking a long time, it seems like lately, but we'll keep our fingers crossed that they come back pretty quick.
Great. Well, that kind of ties into future agenda items or things to keep track of. Any other updates folks want to share out? Or also, if there's future agenda items you would want to flag for us to start exploring, we have a lot of time between now and the fall.
Just noting a couple of comments,
No.
just so folks don't miss them in the chat. Some references about stellar sea lion biology. And Jess, thank you for leading the sea otter charge. I think I noticed in the Pacific Whale Watch Association has put out a summary report the last 2 years, and the 2022 year showed quite a few sightings of sea otters inside the San Juan Islands, as opposed to just at Race Rock. So that's timely and exciting to start thinking about them a bit in the inland waters.
Yeah, and it'll be, technically this year is a year that I'm supposed to update our periodic status review. I'm a little behind with orcas and murrelets at the moment, but so probably 2024 will be update of that state status review. We'll have to kind of reconsider our recovery objectives based on the Jesse Hale paper from last year. So it'll be a fun conversation. I'll need help.
Okay, with the last couple minutes here, just want to ask, pose the question one more time, if there's any future agenda items that members on the call would like to flag for me as we start planning for fall. And we'll be communicating through the listserv. I'll run through the action items. And feel free to reach out to me after the meeting as well with any future agenda item requests. As far as action items, I'm gonna follow up after this meeting, maybe tomorrow, just with a review of the upcoming events that we flagged towards the beginning of the meeting. So everyone has those and has the links. Some are as soon as this weekend or next week, so want to get those out before the meeting summary. And then also in that email, just reiterating, we would love additional review of the orca's vital signs. Probably open it up for another week or so, just acknowledging not everyone had a chance to review and provide feedback. So again, they are updated as of right now on the PSEMP website, but there is flexibility with the timing. So want to put it out again, since this is the version that will go into the state of the sound. And then we'll be following up with a meeting summary in a week or two with a link to the recording. And we'll be following up with folks as I know it'll be a busy summer, excited to hear what you all have done when we reconvene in the fall. But we'll be reaching out in the next few months, just teeing up what the fall meeting has in store. I really appreciate everyone's time today. Thanks to our presenters who are still on the call, and please reach out to me or Scott or Corey, who couldn't make it today, but sends her hellos if you have any questions.
Yeah, special thanks to Hendrik and Tom for those wonderful presentations, and please say hello to Joe for us.
Thank you for the invitation. It was interesting listening in and being here. Thank you.
Thanks, everyone.
Thanks a lot,
Enjoy
everyone. Have a good day.
Bye.
Thanks.
Have a great day. Bye all.
Bye.